Free your mind, but not your experiments. Welcome back to Realizing Research. I am Samantha Deal, a graduate student in developmental biology, and today we're talking about controls, probably the most important topic in all of science. A control is an experiment or an observation designed to minimize all variables other than your independent variable, aka the thing you're interested in. Cool, why is that important? Let me give you a very simple experiment. Let's say you want to test whether or not a fertilizer helps your plants grow better. So you buy two plants. You plant your regular plant without your fertilizer in your backyard, but then you plant the other plant with the fertilizer in your front yard. Well, you can see the issues with this. There may be different sun levels, there may be different wind levels, there may be different water levels, and those are all really important things for plant growth, so you won't be able to really tell if the fertilizer has an effect. The proper control would be another plant right next to your plant that is using fertilizer. It can be very difficult to know exactly what the proper control is, and most of the time when science is brought into question, it's because the proper controls were not used. Let's go through a few examples to explain what I mean. One of the simplest experiments to determine the control is a case where you are using a drug or a physical or electrical manipulation to generate your experimental condition. In this case, the best control is obviously just another animal that doesn't have that physical or electrical or chemical stimulus. There is one caveat to this, and that is when you get outside of the laboratory setting. I mean, when you are testing whether or not a drug works in humans. You cannot regulate humans' environmental conditions, so you have to get around the best control by just increasing the number of samples. This type of control is pretty easy to recognize, so let's talk about something more difficult. I really like my imaginary gene called supercool gene. So let's imagine a scenario where you're studying supercool gene. You have made a mouse that is a mutant for supercool gene, and you want to know if in fact this gene is regulating that mouse's coolness. You probably have plenty of experiments planned, such as determining the mouse's musical interests or testing whether or not it prefers ties or bow ties. These are very important things to test. But what is going to be your control in this process? Because the process of making a mutant can be rather disruptive to DNA, and you could have other mutations in the background, and you likely do. So how do you determine if your gene is the one causing your phenotype? What if you unknowingly have another mutation that is the gene that causes your mouse to choose bluegrass music? There are ways that scientists get around this, one of which is so-called outcrossing, where you try to just eliminate as many other mutations as possible. This technique is commonly used, but I would argue that probably the best control for this scenario is what's called a genetic rescue. So you take your mouse that is mutant for supercool gene, and what you do is you bring back a copy of supercool gene. So you can still have all of these other mutations and other genes. But if your mouse who has a mutation in supercool gene chooses bluegrass, and then your mouse who has a mutation in supercool gene plus has a rescue for supercool gene suddenly switches to smooth jazz, then your gene is likely the cause of that phenotype. So in this case, your genetic rescue is your ideal control. Let's move on to a very, very complicated sample. I'm going to talk about DNA sequencing, and particularly within humans. It's becoming more and more common to do genome or exome sequencing within patients to try and identify what is causing a disease or disorder that they have. How do you determine whether or not a mutation or a variant in the DNA is causing your disease? After you've sequenced the patient, if you find a mutation in a known disease-causing gene, then you have an answer to your question. But if you find a bunch of mutations in other genes and you don't know that they cause disease, then you don't know what is happening. We have a so-called reference genome, and this is the most common DNA sequence within all individuals. As we've sequenced more and more people, we've found that there are a lot of variants within individuals, and it's unlikely that all of these variants are actually causing diseases. So comparing your variant to the reference is not the best case scenario. Okay, so then maybe you can compare your patient's DNA sequence to their parents', right? Children usually have a handful of variants that don't seem to be present in their parents. And it's unlikely that every single one of those variants is causing disease, so your parents are not really the ideal control. Honestly, at this point, there is no ideal control. You need to, at that point, take that variant into the laboratory setting. I mean, you need to generate that mutation within mice or within other model species or within human cells, and you need to see if it does in fact cause a phenotype. You've seen many different types of controls now and cases where there is no real ideal control. Let's talk about one more thing, which is called positive controls. Positive controls are experiments where you know that they have a phenotype. Why is this important? Let me give you an example. I work in a laboratory where we try to find novel modifiers of dopamine. Dopamine is a chemical within your brain that's important for memory and movement and many other things. We have a new tool that's able to measure dopamine. The thing is, you need to make sure that tool is working. So you need to have a positive control where you know it modifies dopamine, and you run that against your negative control and you test whether or not there is actually a change in dopamine. If there isn't, then something is wrong with your tool and you need to step back. Positive controls are really, really important within papers where they're using a new technique. They need to validate that their technique is in fact working. And honestly, scientists are always coming up with new techniques, so they're used a lot. That's all of the type of controls that I'm going to talk about today. I mostly just talked to you guys during this Slice of Science, so if you have any additional questions, please do put them in the comments. And I will see you guys during the next video. Bye-bye. Welcome back, you silly gooses. A control is defined as an experiment or this is a test. This is a test. Today we're testing things. Test it. Testily. Round 3. Foot. Hey, Kit Kat. Kit Kat. It's inside a number. Later, taters. Control.